The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You logged on to the Nelson Labs webinar, Advancing Microbial Contamination Detection Through Rapid Sterility Testing. We just have a few housekeeping items that we'd like to go over before we get started. If you ever missed one of our webinars or simply like to refer back to one, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can receive future notifications of upcoming live and on-demand webinars by liking Nelson Labs on Facebook or on LinkedIn. We also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you will find curated content from industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 900 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. So let's get started. Today we are joined by Zach Anderson and Scott Diamond. Zach's career journey at Nelson Labs began in the bacterial endotoxin lab in June 2000. Over the years, he's transitioned through various technical roles, including lab analyst, study director, and technical specialist. In 2010, he ventured into management, initially overseeing the BET department and later managing multiple lab sections. In 2017, Zach took the role of Director of Laboratory Operations at the Salt Lake City site, eventually assuming site leadership in New Jersey. During his tenure, Zach has gained extensive experience in bench work, management, and leadership across departments supporting sterility assurance, environmental monitoring, packaging, filter testing, microbial IDs, non-sterile product testing, reprocessed medical devices, mycoplasma testing, antimicrobials, analytical chemistry, and more encompassing routine and project workflows. Scott started his career at Nelson Labs in June 1988, when Nelson Labs was only 3 years old. Scott initially started performing bioburden testing as an analyst, learning aseptic technique skills and radiation methods. This testing was followed by bacterial endotoxin testing, mainly using the LAL gel clot method to detect pyrogenicity. Scott, along with others, then helped initiate optical density kinetic methods at Nelson Labs. Over the years, he's transitioned through various roles, including biocompatibility study director, performing thousands of MEM elution and overlay tests. He helped bring up the chromosome aberration test to Nelson Labs. Scott has also assisted in healthcare reprocessing, helping to validate TIR-30 medical device reprocessing tests. He then began his journey as a manager in sterility assurance department as a section leader, where he gained much knowledge of clean room operations, isolator methods, and method suitability testing. Currently, for the last 5 years, Scott has been a Nelson Labs senior scientist, using his unique breadth of scientific knowledge for the Nelson team and sponsors around the world. We'd like to encourage you to submit your questions at any time using the questions box during this presentation. Zach and Scott will try to answer as many as we can in the remaining minutes of our webinar. Now, beginning today's presentation, I'd like to turn the time over to Zach Anderson and Scott Diamond.
Thanks, Mike. So thank you all for joining the webinar today. I appreciate the introduction from Mike, and I appreciate everybody who's joining. Quick overview of the webinar. We're going to talk a little bit about why to consider rapid sterility testing. Are the alternatives acceptable? We'll talk about a path to rapid methods for your product and what is required. And then we'll review a case study, and then hopefully we'll have time for some questions at the end. So you wouldn't know it by looking at us, I hope. But Scott and I, between us, we have 60 years of lab experience. Throughout those years, whether a product is limited shelf life, has some urgent release requirements, whether it's biopharmaceutical, pharmaceutical, compounded, an inventory constrained raw material, in-process samples, or combination devices, really any kind of product. One thing has been constant, and that is sort of the multi-layered and consistent challenge with turnaround time. And within that kind of broad turnaround time category, product sterility has been one of the most impactful tests. I'm certain, with each of your roles, with the people joining on the webinar today, you've all faced turnaround time challenges. And the recovery from that downtime that causes headaches. Unfortunately, with time constraints and pressures that turnaround time put on us, we have to consider the underlying quality impact. If people are, pressured to cut corners, we want to make sure that we have the flexibility and the technology that supports our processes in a way that helps us maintain quality. So considering, some of these familiar scenarios, delayed production, product backorder, lab backlogs, and dare I say an OOS investigation, you can go from a 21 or a 14-day incubation turnaround time, a 21-day general turnaround time, kind of on the basis of a 14-day incubation. This can turn into really a 2-month ordeal in some cases. And so what we really want to talk about today is the benefits of rapid methods to help product availability and ultimately patient safety. First, is the alternate method acceptable? The easy answer is yes.
Hey, Zach, I'm not seeing the slides advance, so I just want to make sure that the audience can see the slides advance.
Are we having technical difficulties already? Are you seeing them now?
No, we're still on the slide, on the title slide.
I'm able to progress the slides if you want me to do that.
Yeah, they're progressing on my screen. Let me unshare and then share my screen again, maybe. Let me know if that changed.
That is great. Thank you.
Okay, you can see it now? Okay, apologies. So let's restart. Well, there are many references in support of alternative methods. We're going to really focus on one today, and that's USP 1223. In USP 1223, there's an acknowledgement of some of the limitations of the current methods. I've listed a couple here. The actual limits of detection of compendial microbiological methods have never been established quantitatively. And the CFU as an estimate with a variety of limitations is prone to underestimate the number of cells, right? So we already know that our methods are imperfect. USP also provides support, kind of understanding those limitations. It's got statements in General Notices 6.30 that allow for, the application of alternative methods. And there are some guardrails, right? So as we qualify or validate alternative methods, we have to kind of understand what the rules are around that. All of the methods and procedures below 1,000 are considered referee tests. So in the event of a dispute, regardless of the application of an alternative method, you would revert back to the referee test, in this case, USP 71. And There's also some guidance provided on, how to navigate the compendia requirements. Once you qualify USP 1223, you're still going to need to understand, where your product is marketed and what the compendia in those regions or those regulatory bodies may expect from your product. I've also included a list of useful references for background. We won't cover these today, but they're listed in USP 1223. And I included PDA technical report number 33 as another very useful reference as well. Okay, now that we're generally oriented, what's next? As you see, there's a fairly well-defined pathway provided by USP 1223. There are some familiar elements in this process, such as establishing your URS, selecting the right technology for you and your product, evaluating the key factors that may impact your process or your product, demonstrating the applicability of your chosen technology and method, and then validating your method, demonstrating your product's suitability, and in this case, the test article effects. And then you can celebrate with your newly acquired rapid test capabilities. And like I said, some of the elements are very familiar, I'm sure. Instrument qualification, your IQ, OQ, PQ, validating your alternative method, showing that it performs equivalently. This is generally established by the vendor, but it must be confirmed by the user. Your method suitability, it must be, the technology must be suitable for your specified test. This is similar to your BF, but in the case of qualifying and validating an alternate method, it's much more fun. There's quite a bit more data gathering. And then there may be some processes that you're less familiar with if you've never established an alternative method. So first, understanding the limitations of the current measurements. The USP 71 test is a qualitative method, but understanding sort of all of the elements that go into the test, understanding the limitations of the CFU, and then working on demonstrating, your equivalence. And we'll talk a little bit more about that. And then application of a non-inferiority concept for the method validation. And then because of that approach, looking at the guidance on statistical methods. Understanding the data. USP 1223 gives guidance on how to demonstrate equivalence with 4 options: acceptable procedures, performance equivalence, results equivalence, and decision equivalence. Now, remember, 1223 is not only utilized for sterility testing. Knowing your application is important to the correct approach for demonstrating equivalence. And if this is your first exposure to the concept of non-inferiority, it's the demonstration of that the alternative method is not worse than the compendial method by more than a pre-specified amount. And that pre-specified amount we'll talk about a little bit later. With that approach, there are a couple of statistical methods used for the qualitative approach. Both use the non-inferiority hypothesis. USP 1223 outlines approach number 1, which is to demonstrate decision equivalence, and then approach number 2, which is converting qualitative data to quantitative results using an MPN method. So where we have the USP 71 being a qualitative, sort of binary test, did it grow or did it not grow? That's where we'll get into our decision making. And you can see below in the table, which is an example from 1223, there of the 4 options, 3 of them require comparison to the compendial method. The acceptable procedures is the one that doesn't. Now, while there are several steps for method validation, overall the process is fairly simple for the user. It's a standard sterility test. The incubation is roughly 6 days. The assay is aliquoted. If microbial growth occurs, the elevated ATP will be extracted and amplified. That reaction produces photons of light measured in relative light units. and then the results are calculated. So it's really an enhancement of the current test process. So, in terms of, the leap from the classical method to the alternative method in this case is fairly simple, aside from, the technology itself. And the simplicity, once all of the validation requirements are met, leads to a handful of benefits. It's applicable to a broad range of products. It's established technology. Now you've got a validated method. You've demonstrated compliance, and your results are achieved in half the typical 14-day incubation period. So one additional added benefit that we've already seen is the potential to reduce interfering factor caused delays. If you've got a non-filterable product, or you have colored or opaque products, or neutralizer media that appears turbid, this technology can help support faster results, because you don't have those subjective visual cues. And you can see below in the graph, regardless of subculture requirements or verification testing, additional verification testing, you're looking at it still within that 4 to 7 day window for testing and release. Okay, that's my part. I'll hand this over to Scott Diamond. He's going to talk through a case study that hopefully will really show the process of qualifying and validating these test methods. Again, if we have any questions, go ahead and enter them in the chat, and we'll get to those. Thank you.
Okay, I will start this out by going into the case study. This was kind of the fun part for me, because I got a lot of hands-on experience with performing a lot of this testing for rapid sterility. So with this case study, I really want to look at the performance qualification and how that works, since this is really kind of the longest lead time part of the testing, and it also can give us a lot of information for how the test sample is working with the rapid sterility method. So basically, with the performance qualification, we are able to start the testing by how we started the testing was we had a partnership with Nelson Labs and Charles River. This was very key to our success in having someone that we could work with and develop these tests within our laboratory facility. Our test article that we chose, or that we were able to obtain, was a 0.9% sodium chloride solution that's used with a dialysis treatment. This is found in type of dialysis bag is how that comes to us. Once that performance qualification testing was performed at the Charles River location, we needed to perform a transfer of the performance qualification to the Nelson Laboratories Salt Lake facility. Basically, what we're wanting to do is our goal is to verify that the similar results would be seen at Nelson Laboratories that we're seeing at the Charles River facility. But before we even we can start the rapid sterility method, we have to understand, would the sample cause interference to the test method? And so we perform a sample effects test. The sample effects test basically evaluates and assesses the compatibility and the feasibility of the test article with the rapid sterility test system. We are trying to make sure that the test article is not inhibitory, or would cause, basically, if there were organisms present, that they would not be seen. Also, another part of this test is to make sure that we don't have a test article that already contains background interference that would interrupt or interfere with us being able to see the appropriate results. As part of this sample effects test, we have some very key controls and test samples that we need to test. The first one is the broth calibrator. That's performed with either the soybean casein digest agar or the thiol or fluid thiol glycolate media, which is for anaerobic organisms, where the soy casein digest broth is for aerobic organisms. This becomes our baseline for the rest of the tests. So basically, whatever we see in this media, that is where we're going to start with the other tests. So we need to use the same lot and media type as we will be used for the test samples. Next control I'll talk about is adenosine triphosphate, or ATP control. Basically, this is adding ATP to the media so that if ATP was available or present, we would be able to detect that. As you look to the right of the screen, you can see that our ATP positive growth should show positive control or positive results, basically for the presence, or that basically a positive result was there. The microbial broth control is similar to the ATP broth control, but we're using an actual organism that we've selected to spike into our media and to determine that we could actually see growth if it was present. And in this case, we did see growth. The control product sample, or we call CPS, is basically just the product itself with a growth media in it. No ATP or no organism spiked here. So what we would hope is that we have a sterile product and therefore the results were 0. And in this case, we did see that no growth was present. The control product sample ATP positive control is another control that helps us know that if ATP was present in the sample, we would be able to detect it. So the ATP is spiked into the media and with the sample, and we are hoping that we would detect that ATP, and in this case, as we see on the right side of the table, we did see a positive result for that. The last control that I would talk about is the control product sample microbial control, where we take that selected organism that we used in the media and we now spike that with the test sample and the media, and we are hoping again that we would see no interference or that we would see a positive result, and we did so within our case study. This slides how we set up the performance qualification for the dialysis solution. There's a lot going on here in this slide, and basically with the USP 71 media, we test both the soy broth, TSB, tryptic soy broth, and the fluid thioglycolate, FTM, and we are able to see within just the USP that we would, again, have no growth, and that's part of that broth calibrator that we talked about. In the USP 71, the preferred method for testing sterility is a membrane filtration type of test. And I'll explain this method as we look at what we consider the closed membrane filtration using canister sets. The canister sets are designed to basically have a system where the product can go through the system and the tubing and be drawn into these canisters and be filtered through a membrane that is within inside these canisters. Basically, the fluid will run through these and be filtered, and any microbial content would be contained on the top side of the filter. The peristaltic pump that you see moving here is drawing that solution from the bag that we have here that has the dialysis fluid, and it pulls it into those canisters. We can control how much fluid goes into which canister by some clamps, and so we basically can select which canister we are filling and know that we can do those separately. One could be a soy media control and one could be the thio in that case. For this particular example, we were actually trying to get replicates of the same sample, so we would pull that into the different canisters that we have here. The first one would be a broth calibrator. that we talked about earlier that is used, and we have a rinse solution that would go into that and rinse anything away if it was present, and then a sample itself showing that the sample itself would be negative. The bottom part of this screen shows us that, maybe I'll use my laser pointer here, that we have 3 different other sets of canister sets that we're doing for replicates. Basically, this one that's on the top has 6 different canisters, and that's for our 1 CFU dilution. So we will take a microorganism suspension. We would dilute it to what we would consider would be about 1 CFU, so a very small amount of colonies of the organism would be present in this 1 CFU type of test, and we have 6 going on there. What our hope is that even with low levels, even if 1 CFU is present, we would be able to see growth. The next set of controls down here are our 10 CFU controls. So this is one where we have a little larger population of the organism, and we're hoping again that we will be able to see that. And these 4 different replicates for each organism will be tested for this 10 CFU. The bottom set that we have here is for the 0.1 CFU. Again, when you test 0.1 CFU, you're not expecting to see a lot of growth. This is just according to the alternative method, we can again be monitoring our system and statistically looking at what our results will be. We also, just to kind of conclude, that vacuum pump will actually pull the liquid through, so the test sample liquid will go through that membrane and be pulled out and discarded, where the contents on top of the membrane will be tested. This table shows that the scenario where we are taking the USP 71 organisms, and we're doing that for both the fluid thioglycolate media for anaerobic organisms, and we're also looking for the tryptic soy broth media, which is aerobic media. These different organisms will be tested in the FTM that are in the top part of the table, and the ones below will be tested in the TSB. The one thing that we also will be testing is environmental monitoring isolates. We, with the particular case study, we selected 2 isolates from the manufacturer of the sodium chloride and what their process is. So they basically sent them their organisms to us so that we could create a suspension and have that tested at these levels that we've talked about. We also tested an environmental organism from our laboratory as well as part of this testing. So the temperatures are shown here for incubation, and then at 6 days of that normal 14-day incubation, we're going to go to our next step, which we'll see next. Basically, this screen helps show the key factors that are seen in rapid sterility testing. Basically, we need to show a limit of detection. This is basically the lowest number of microorganisms in a defined volume of sample that can be detected, but not necessarily quantified under the stated experiment. The equivalency, another key part, is to show the statistical equivalence between the rapid sterility test and the compendial method, and gather all the information from all of the inoculated canisters and basically demonstrate that we have equivalence between the 2 different methods. And the specificity is the ability to detect a range of microorganisms, which demonstrates the method is fit for the intended use. And again, just recapping, we do take many different types of organisms, the 6 USP, the 3 environmental, the 1 slow-growing organism is also performed to basically show that if there was a scenario where an organism was a little bit slower in growing, that we would still be able to detect it at that 6-day period of time. And we also stress organisms, and one of the organisms we typically stress is the slow-growing organism, and say if there was anything that harm, that organism wasn't totally healthy and maybe took a little longer to grow, that we would be able to see it. And again, we're performing that in concentrations of 10 CFU, 1 CFU, and 0.1 CFU. We, of course, run positive and negative controls to show everything's working correctly. We run side by side this method with the compendial method, and then we will look at those statistical analysis. During the 6th day, we basically will get a lot of canisters now that have incubated and have either are showing growth or they might not be showing growth, and this is part of what we're doing. In this particular test on day 6, we removed an aliquot from these canisters. So we make a homogeneous solution so that we can make sure that we have captured anything that's within that canister, and we have those prepared for the Celsis testing. On the 6th day, we also perform visual scores for each of the samples, and we're going to score these growth or no growth. So in this particular example, this one on the left was very turbid, and we were able to easily show that this was a positive sample. It was growing, it was showing turbidity. These are normal characteristics for a sterility test when we have organisms present. And this was actually our 10 CFU canister showing here. The one on the right was actually our broth calibrator, which is just the broth media. It should not show any positive results, and it did not. We have also our sample, which we are hoping would not have any positive results, and it did not. We had our 0.1 CFU canister, which could have positive results, but in this case, it was not. We didn't see very many of those during our testing. And then we saw one actually that is also on the right, a little larger image that had a look. It's hard to see it, but you might take a look that there are little specks in this media, and it's difficult to tell if that was growth. Especially, if we see the one on the very left up in the other picture, we're definitely not seeing the turbidity that we saw this was actually after 14 days. So that during the 6 days, you just don't, a lot of them look very much like this one that we're seeing here. So that's our question mark. My question to you would be, do we call this a positive result? Do we call it a questionable, that we're unsure? Do we get a bunch of Nelson Labs scientists together and vote on whether we call this a positive or a negative, or that we're unsure? So this was kind of our question with this particular study. This was kind of the cool part, that we learned a little bit of things from this particular test. And to understand what's happening with the Celsis testing is shown in this diagram in the slide here. The Celsis Advance II system that we use, that we purchased from Charles River, and it is designed to detect and quickly detect if there are microorganisms by looking at the enzyme-enhanced ATP bioluminescence. ATP is essential for the energy of all organisms, and it is targeted by this particular test method. When present, microbial ATP triggers a reaction within the glucarase enzyme producing light. Down here in this picture below is a picture of the instrument itself with these reagents that are added to the test, to these samples that we've taken and we're testing in duplicate. Basically, one of the first reagents will extract the ATP from the cell, and with this, we are able to put in a second reagent which amplifies it. This picture down here is showing that situation where we just have very little or even just one ATP that is being extracted, and then the amplification generates a bunch more ATP. That's going to help us be able to see that, visibly see that in a test on the instrument, on the rapid sterility testing instrument, and be able to show that there was indeed a high level of microorganism present here. I do want to just go through, this was kind of part of the learning and kind of the fun part of the test as well, where we did see some interesting things as we were performing this test. The first thing is that we were able to, and this was actually the slow growing organism, so it kind of worked out well for what we were trying to show with that organism. At 6 days, the visual score for this replicate 1 showed that we scored it positive, but the rapid sterility results showed negative. So that was something that caught our eye. We also saw, in this case, the 6-day visual score, we would have scored as negative, but the Celsis scored it as positive. And the replicate number 6, we saw at 6 days a score of no growth, but the rapid test showed that it was positive. So those are some things that we needed to understand get a little bit better understanding. We did 7-day results, which are typical of our sterility testing, where you test halfway through the sterility test of 14 days, we do a 7-day sterility test, and we scored those at 7 days. And I believe because we were agitating these and we were taking aliquots, that we kind of stirred things up, and it looked like everything was growing, all 6 replicates at this 1 CFU. At one time, at the 6 days, we actually take what we call subculture from those aliquots, and we put that subculture portion of the media also on agar plates. Once those were incubated and allowed to show growth, we actually saw some very interesting results. The replicate 1 actually showed 0 subculture growth, which very much corresponded to our rapid sterility results. So that was a clue that something was going on here, that with our rapid sterility results and our subculture results seem to be matching up. The next thing I did is we waited for that whole 14-day incubation, and we performed an investigation. The investigation was basically running the test samples again, taking new aliquots from the canisters, and running them again on the rapid sterility test system. We were able to also see that the same results were seen as we initially saw at 6 days, so they were very agreeable there. When you have a sample that you're unsure about, like we described with this 1 replicate, replicate 1, we do what we call a media transfer. This also could be called a subculture. And we place the aliquot back into fresh sterility media to make sure if there was an organism present, we would have a chance to see growth. We actually did this in both the thioglycolate media, so anaerobic, and for the aerobic media. And so we were hoping that if there was an organism that was having a hard time growing in one media, but the other media, we would be able to see growth there. But no growth occurred. So that basically helped us know what that first replicate results were actually not organism related, but the precipitate was something else that was going on. Sometimes you see that in sterility testing. It's not always the case, but it is something that can happen. And you can imagine that when we're testing sterility samples for a sponsor, when we have a scenario like this, the same question can come up. Do we score at growth or no growth? The kind of take home, I feel like, message here is we learned that actually at 6 days, we were seeing the actual results with doing the rapid sterility testing. When we want to look at the results that we had for all of the organisms, we talked about the 10 different organisms that we selected to test for this particular product. We were able to see, and with the Charles River data, that there was kind of a trend that we could see, or a pattern that we could see to the different results. One of the first patterns that we saw was here at that 0.1 CFU level. You're not seeing a lot of positive results, just a couple across the board here. And so basically, we're not going to see a high level of organisms growing when we're diluting that to these low levels. So that was pretty much normally what we would expect to see when we had that transfer method in-house, which of course we saw the same type of results. We also can look at the 1 CFU inoculated or diluted inoculation in these canisters. We see a lot more positives popping up here. So a lot higher probability that we're going to see 1 CFU. Those ones that are showing negatives are not a scenario where they're not detecting CFU, but just that there's a window where some of the canisters will have organisms and some of them won't. So the ones that have the organisms will continue to grow, and we want to look at that in comparison with the compendial method at 14 days. Last thing we'll look at is the data that we saw with the 10 CFU. You can see across the board we have all positives for all 10 organisms, which indicate basically 100% recovery when we're spiking at that level. Again, very low levels. Typically when you run a BF test, bacteriostasis, fungus stasis test, that is, a method suitability test, you are inoculating with less than 100 CFU. So you can see in comparison, we're using a much lower amount of inoculum. Looking at this data on a bar graph can also kind of take home those same type of points and maybe give us even a little bit more learning. These are those 1 to 10 dilutions that were done for the 0.1 CFU here. Here is a situation where actually one of the organisms were showing a little different result compared to the compendial method to the Celsis method. And so with those results, that's something that we can get learning from, and we'll be able to take that data and look across all of the organisms and statistically calculate, if this is a factor, is this going to be an issue or not. I just wanted to point out here with this particular slide that all of the organisms for the replicates, all the replicates I should say, for the 1 CFU grew. With our 1 CFU inoculum, these bar graphs are showing that column. Not all of them grew in all 6 replicates, which we kind of saw in that table of results, but these actually will have scenarios where, again, some grow and some don't. With the replicates that we said were 100% at the 10 CFU, those were only 4 replicates. So even though those bar graphs aren't going up to as high as this one, they're still showing, like I say, 100% that they were growing. So this data was very interesting and was able to give us a lot of information and be able to use for our statistics. Zach's going to talk a little bit here about those statistics.
Thanks, Scott. That was great. So as we look at the statistical calculations, you take all of that data, and like I said before, we're gonna use approach 1 as outlined in 1223, which is demonstrating decision equivalence. So for each organism, we calculated the detection probability at 1 CFU inoculum level. We used a fairly simple statistical equation, the number of positive replicates divided by the total replicates. We used a non-inferiority margin of 0.2 or 20%, and that was that pre-specified amount that was described before. The non-inferiority test was evaluated using 95% confidence intervals, one-sided, to evaluate the difference in the detection probabilities between the 2 methods. And so ultimately, the method was validated as non-inferior, demonstrating that we can establish our decision equivalence. And some of the key learnings, again, the rapid sterility is a great alternative for many products. It greatly reduces turnaround time. The detection using ATP bioluminescence can help reduce the chance of subjective false positives. And then once the limit of detection equivalency validation is complete, we can, using a specific method, whether it's direct immersion or membrane filtration, we can then perform suitability assays on different product types. And sort of to close, Nelson Labs provides not only the guidance to transition from your existing method to the Celsis rapid sterility method, We also provide ongoing support with developing and executing the testing, facilitating the adoption, and performing routine testing. We're committed to continuous support, ongoing test services to help maintain and your compliance and sterility assurance, with support across the board for your sterility assurance programs, not just the sterility methods. And with that, I'll turn the time back over to Mike for facilitating the question and answers.
Thank you so much, Zach and Scott, for all of your insights. We've all learned some great information today, and you've given us all a lot to think about. We do have a few questions to come in, and we do also encourage you to submit. If you haven't submitted already, please do submit your questions. If we don't get to yours, I'm sure Zach or Scott would be happy to address it individually after we conclude. So we do have one question in here, and the question is, what is the estimated number of individual samples needed to perform the performance qualification testing?
Scott, do you want to take that one?
Sure. For this particular assay that I performed, we tested individual samples. So the amount of samples needed will be a little different if you have to pull samples. Let's say some sterility test will pull 10 samples together. We need to show basically those 10 samples for each type of organism. But for this particular one, we were one basically sample per each test. So for the performance qualification, that was about 150 samples that were needed. When we add the sample, the method suitability samples, that's going to be about 12 more. For that sample effects test that I said, that we're looking to see if something is going to be inhibitory or interfere with the study, we'd like to at least have 2 samples for that. And then for just to make sure that if something, we have to redo a test or anything, that we have about 10% more. So we had about 180 samples that we required for that particular test.
Great. Thank you, Scott, for that excellent answer. We've had lots more questions come in, so I'll turn, this will be open to either one of you as well. The next question is, what do you do with out-of-spec samples that do not visually look positive? Do you continue the incubation?
So I'll go ahead and start if that's okay, Scott. An OOS will follow the same path as a general OOS, right? So I would say you continue the incubation as long as the sample's been handled in a way that it wouldn't be compromised in any way to continue that incubation. You wouldn't want to perpetuate a false positive. But from there, I think it follows the same phase 1 laboratory investigation as any other OOS. So whatever the investigation road leads you down, the identification of the organism, the evaluation of the testing, whether or not there's videos to review, looking for aseptic technique or any chance of initial contamination, and if the sample is I guess determined to be appropriate to continue the incubation, you may gain additional information from that process as well. The good news is you have an early indicator. 6 days or whatever the validated timeline would be, you can kind of make the assessment there, retain the sample preparation, and utilize that as best you can in the investigation. I don't know if you want to add anything to that, Scott.
Yeah, I think that all that information was great, Zach. I think the cool thing that we learned about the rapid sterility testing is those 6-day results. You can see things there with the ATP that may be visually scored as a positive type of result. So really we're able to take away those kind of questionable samples and understand the data for that. We we have confidence because we do the validation up front with the test samples, and we can see that through the validation, through the method suitability, that we're not going to score something incorrectly with the Celsis system. But I agree totally with what you were saying, Zach.
Yeah, and I would also add maybe to that, the method in and of itself doesn't entirely eliminate some of the challenges that you'll face with product sterility testing. So, you know, it's very wise and really expected to have a full quality control process within your sterility testing. So, you know, it shouldn't be the only layer of defense to be able to re-incubate. You've got to have, like I said, maybe video cameras, maybe robust validations around all of your cleaning methods, your cycling, your processing, your handling, your aseptic technique training, you know, just the whole host of items that are traceable within an investigation that will help support any decision that's made.
Great. Thank you guys for that excellent answer. Our next question is, what types of products is this good for? This system, I am assuming.
Yeah, initially, you know, it makes sense to use for short shelf life products where you're producing a product and then releasing it either at risk or, you know, there's some urgency around the release. But I would think as these technologies become more supported and more accepted, I think that range of acceptable products becomes wider. And I would fully expect that at some point, the majority, if not all, sterility testing will be performed, you know, with these molecular or rapid methods.
Great. Thank you, Zach. We'll go on to our next question. And the next one is, what is the maximum number of samples that can be tested in one assay? I'll open that up to either to Zach or Scott.
Okay, we do test in duplicate, and so there are basically 162 different wells that can be tested. You know, some of those are going to be controls. So you also, if you have different media types, you need to put spaces in between those samples. So really, you can perform, you know, much over 100 samples if they're all in the same type of media. And so a lot of samples can be scored and be run on this this type of test.
Great, thanks, Scott. We have a little bit more time. The next question is, do we perform a hold time study, and what's the longest hold time, if so?
Do we hold time study for retention of a tested sample, or maybe we need a little bit of clarity on that question?
Yeah, sorry, that's the information I do have. If we get clarity, we'll come back to that one. So here's the next question. This person was a little late to the beginning, but their question is this only available at the Charles River facility, this type of testing? And would this typically be recommended by the lab depending on the product samples, or is this something that the customers ask for?
Yeah, I can probably take that. So Nelson Labs has this capability in Salt Lake City. We're also implementing the capability in our Wiesbaden, Germany lab as well. There's opportunity to place it in other centers of excellence, which we intend to do. So it's not only available through Charles River. In fact, you know, we've partnered well to get this technology online, and we'll continue to do so as it supports our clients. I would say, you know, there may be client demand for it. A client can ask for this capability or this test method from Nelson Labs, but also, you know, I would encourage people to explore this technology for some of the reasons that we described in the flexibility that it provides, not only for inventory management, but also recovery time and the flexibility in turnaround time overall. You know, the 14 day turnaround time in difficult situations can turn into, longer ordeals that disrupt essentially everything that we do when dealing with sterile products. So, I would really encourage people to explore this technology, and we're more than happy to help with that.
Thank you, Zach. So we do have a clarification on that previous question. That was about if we perform hold time studies and what's the longest hold time. The clarification is the retention of the sample.
We haven't necessarily that I'm aware of, Scott, but the test itself is not necessarily different than the existing process. So conceivably, you would hold it for 21 plus days, if you're performing an investigation. And at that point, you would have to look at, what it meant for the sample. You could perform a hold time study, but generally when we're testing product sterility samples, we will perform the testing, we'll confirm the results, and then we have a, maybe a very short retention period before the samples are discarded. And a lot of the time it's simply due to space constraints. If we have high volumes of completed tests, we wouldn't have space to hold all of those samples. So I think it would be we still have that flexibility in terms of the general classical method taking longer than this method if you needed to retain a sample. Hopefully that answers that question.
All right, yep, thank you, Zach. We're going to try to squeeze in one or two more. Next question is, does this only apply to a liquid sample, or could this also be applied to a physical medical device?
Scott, you may have a different opinion. I'll let you go first.
Yeah, and you can basically, yeah, the method that for rapid sterility can be performed with different methods. One of course is the membrane filtration, which we kind of highlighted today. That tends to be for that liquid sample. We can also perform a Celsis study with the same type of process. Basically, at that 6-day incubation period of time, we again remove aliquots, and that might be a little harder to, depending on the type of sample that we have, the type of jar or media. We test in gallon sizes, we test in graduated cylinders sometimes. So really depending on the type of size of the product, we could still do the same test. We're basically just removing that aliquot media from at that 6-day period of time and running that on the Celsis.
Yeah, I agree with that. I think some of the challenges would be agitating to get a uniform aliquot, which in many cases we can solve that problem, right? So I'm a big proponent of evaluating the technology, across the board wherever it may be beneficial to clients and patients.
Great, thank you. We're going to do one more question and then we'll wrap. So this question is this only done with the closed filtration system, or can it also be done with the USP immersion method?
And that's probably pretty much a very similar answer that we just gave. Is yes, that can be done with that immersion, that direct transfer type of right into the media. Again, we would incubate it for the type of media that it needs to be incubated in, whether it needs neutralizers or just the media itself without neutralizers. We can test that for incubate that at that 6 days. Take the aliquots, as Zach's indicated. We have to make sure we have a homogeneous type of solution, and then we can take that and test it on the instrument for the rapid sterility.
Great, thank you, Scott. Well, that wraps up our time today, and I'd like to thank Zach and Scott for this presentation, their answers to all the questions. We'd like to thank you, the audience, for attending this session, and we hope that you found this to be a valuable experience. Again, we encourage you to reach out to Nelson Labs either at our website at nelsonlabs.com, or you can reach us at sales@nelsonlabs.com for any further questions. You can also reach out to Zach and Scott as well. Again, we'd like to thank you, the audience, for attending, and we hope you have a great rest of your day.
Thanks all.
Thank you.